Neurons are excitable cells that transmit electrical signals. They are long-lived and have a high metabolic rate. So long-lived means we don't, unlike other cells, we don't keep replacing them through mitosis. We form them early on in our life and they last with us, as long as their lifespan. And once a neuron dies, it cannot be replaced. So neurons are what we refer to as amitotic or postmitotic, meaning they're done undergoing mitosis. All right, they can no longer divide. So basically, the neurons you have now are the neurons you're going to have the rest of your life. So you really want to protect and cherish your neurons. Um, Neuron structure is very important, so we're not going to rush through the structure of a neuron. We want to pay particular attention to this drawing. Neurons have basically what we call the receptive end. So that's where it'll receive a signal or a stimulus. So neurons detect things, and then they transmit action potentials, and then they release neurotransmitter that causes another cell to either be excited or perform some action. So the basic parts of a neuron include the cell body. The cell body is where you're going to find a nucleus. All right, so in this drawing, this is the cell body. There's the nucleus. In this real life neuron, there's the cell body, and you can see a nucleus. Sticking off of the cell body, there's usually some shorter processes called dendrites. And dendrites are where you're going to find this neuron's receptors. So whatever stimulus a neuron can detect, that occurs at the dendrites. All right. And then typically they're going to have a long process sticking off the cell body. It may or may not be myelinated. In this case, this drawing shows Schwann cells that are producing myelin, all right, along the axon. Then at the very end of the axon, you're going to have these little presynaptic terminals, or we called them synaptic end bulbs when we learned about the neuromuscular junction. All right, so if this was a somatic motor neuron, these would all be neuromuscular junctions, for example, this drawing here. But this neuron might not stimulate skeletal muscle, it might do something else. Neurons will have what we call a trigger zone at kind of the beginning of the axon. And that's where the action potential will begin. So this neuron will kind of sense its environment, sense the stimulus, and then it may or may not produce an action potential that travels down the axon, eventually leading to the release of neurotransmitter. You'll notice in between the Schwann cells, there are these little gaps. Those are called nodes of Ranvier. And when we talk more about myelination, we're going to see that the action potentials will actually jump over the Schwann cells, and that way they can get down to the end of the axon much quicker. They require a lot of energy, so neurons are going to need ATP production to perform their actions, and therefore they're going to need a lot of oxygen and glucose. Because of this, they tend to be the first ones affected by cardiovascular problems. So if the cardiovascular system is not delivering enough oxygen and glucose, you typically get a result in the brain first. And these neurons are very sensitive to that lack of oxygen and glucose. So we're going to take a kind of a look at these components again, introduce a little bit of terminology that gets utilized quite a bit throughout the rest of the semester. So you're going to see a lot of these terms pop up again. First of all, cell body or soma. We talked about that is the area where you're going to find a nucleus. Neurons also have a very well-developed Golgi apparatus that kind of surrounds that nucleus. They are mainly going to be located in the CNS, unless you're a sensory neuron. They tend to be found outside of the CNS in what's called the dorsal root ganglia. Now, this is not going to make sense until we get to the spinal cord. So we'll learn more about what this dorsal root ganglia is in the spinal cord chapter. Where you find a lot of cell bodies in the CNS, they tend to cluster together, and those are called nuclei. Now, that's a different nuclei than the nucleus of a cell. It's basically areas where, because you have a lot of cell bodies, there's very little myelin. The myelination is only found on the axons of neurons. And we'll talk more about this later, but when you have a lot of myelination, we call that white matter, because the myelin actually is a white fatty material. So when you have a lot of the cell bodies clustered together, you're not going to have much white matter. So it's kind of these areas of gray matter, and those are called nuclei. When you have a lot of cell bodies kind of clumped together in the PNS, so kind of in a nerve, it makes the nerve look swollen a little bit, and that's called a ganglion or ganglia. So ganglia, again, are going to be in the PNS, where you tend to find a lot of cell bodies. And because cell bodies are kind of the bigger part of the cell, it makes the nerve look kind of swollen in that area. Dendrites, those are going to be the short, tapered processes that stick off the cell body. They are going to be the receptive portion of the neuron. So that's where the neuron is going to have receptors. And if those receptors are getting stimulated, it'll start to produce an electrical change along the dendrites, which we call graded potentials. And depending on what these graded potentials are like, it might trigger an action potential. So we'll talk a lot more about that moving forward. Typically, if you're going to be stimulated, you have to have some sort of receptor. And quite often those receptors are either ligand-gated channels or mechanically gated channels. And we'll talk more about what this means, but a ligand is a chemical. So if this neuron is getting stimulated by a chemical, its receptor is going to be a ligand-gated channel. If it's some sort of mechanical stimulus, like pressure, touch, stretching is mechanical, the receptors are mechanically gated channels. Then you have the axon. That's going to be the long process. It's going to extend all the way to the effector. The effector is whatever that neuron is stimulating or controlling. So in case of a neuromuscular junction, the effector is a skeletal muscle cell. Effectors could be other neurons. Effectors could be other types of muscle, skeletal muscle, cardiac muscle. Effectors could be glands. Maybe it's a sweat gland, and the neuron is stimulating the sweat gland. So the axon carries the nerve impulse away from the cell body to the end bulb. At the end bulb, you're going to find the synaptic vesicles, and then the synaptic vesicles exocytose and stimulate the effector. All along the axon, you're going to have voltage-gated channels. Remember, an action potential is a voltage change, and that change is occurring because you have voltage-gated ion channels. You also have a lot of what's known as sodium-potassium pumps. This is going to be a really important concept because for neurons to be able to do what neurons do, they have to load up the inside of the cell with potassium and pump a bunch of sodium outside of the neuron. All right, and we'll talk more about why that needs to occur. The axon hillock is the cone-shaped area that kind of extends off the cell body and then forms the axon. And within that axon hillock, wherever the action potential begins or starts, that's called the initial segment. And you can refer back to the previous picture to see where those are found on that drawing. We also have names for where you have a lot of the axons. So you tend to have a lot of neurons doing similar things. You don't just have one neuron kind of in isolation. And when you're in the CNS and you have a whole bunch of axons kind of going to the same place, we call those tracks. And then if you have a whole bunch of axons going to the same place in the PNS, we call those nerves. All right, so again, a lot of terminology on this slide, but as we move forward and progress through the nervous system, we're going to be using a lot of this terminology. There are structural categories for neurons. They don't all look the same. And they're classified based on how many things stick off of the cell body. All right, so you'll notice these are the three basic structures. You have multipolar, so multi means many. So if you look at the cell body, you have many dendrites sticking off the cell body. And then you have one axon. So this is a multipolar neuron. Typically, these are going to be motor neurons. So they're going to kind of extend from the CNS and control something. Or they're found completely within the CNS, and they're known as interneurons. The bipolar neurons have two poles, or two things sticking off the cell body. One of them contains their dendrites, and the other one is the axon. All right, so they have two things sticking off the cell body, so they're called bipolar neurons. These are very common in some of our special senses, like our sense of smell, olfactory epithelium. So your olfactory neurons that detect chemicals are bipolar, and then the retina has a lot of bipolar neurons. And then you have unipolar. You'll notice there's just one thing that sticks off the cell body, but then it tends to split, and at one end you have the dendrites, and then at the other end you have the synaptic end bulbs. And these are typically going to be your sensory neurons. All right, so they're going to detect something and then transmit the signal towards the CNS. So these are the three structures of neurons that are found throughout the body. Okay, we're going to start to get into how these neurons work. The journey is not going to be easy. It's going to be pretty tricky. So we got to put pieces together here, and the first pieces we need to learn about are what we mean by ion channels and the different types of ion channels. So an ion channel is simply some sort of protein in the plasma membrane that allows a certain type of ion or different types of ions to either enter the neuron or exit the neuron. And these channels may or may not be stimulated to open and close. If they don't need to be stimulated, we call these leakage channels. And leakage channels are not going to be that important for us. They're just channels that do not need some sort of stimulus to open them. All right, they're kind of randomly open and close, and they might let a particular ion move through the membrane. So for example, this shows a potassium leak channel. So it just is allowing this potassium ion to move out of the cell. If it's a chemical stimulus that opens the channel, we call that a ligand gated. A ligand, or some people pronounce it ligand, is a chemical. So notice this is the channel here. It's closed. This little red pyramid-shaped structure is some sort of chemical. In this case, they're saying it's a neurotransmitter, acetylcholine. And when acetylcholine attaches, it opened the channel, and it's moving, you know, whatever these guys are. It's moving, looks like calcium and sodium through the channel. All right? So the key here is to remember ligand means chemical. And usually that chemical is a neurotransmitter. So when we were studying the neuromuscular junction, the acetylcholine receptors on the skeletal muscle, those were ligand-gated channels. All right, you could have a mechanical stimulus. So some sort of mechanical stimulus opens the channel and lets the ion through. So mechanical might be some sort of pressure on the sensory neurons. So sensory neurons that detect pressure, or touch receptors, A mechanical stimulus could be stretching of an organ that kind of stimulates neurons. And then we have voltage gated. And voltage gated channels open when the voltage changes. So in this panel, it shows that there's a voltage of negative 70 millivolts. And then because that changed to negative 50, it opened this channel. All right. So voltage gated channels are going to be very important during the formation of an action potential. All right. Okay, so that just introduced us to different types of ion channels. There's another type of channel called the sodium-potassium pump. And this is an extremely important ion channel to understand. All right. Its job is to produce or establish sodium and potassium gradients across the plasma membrane. So what does that mean? A gradient is when you have a lot of something in one place and little of it in another place. And in this case, we're talking about inside versus outside of a cell. So what does the sodium-potassium pump do? First of all, it requires energy. So this is one reason why neurons require so much energy. They have to constantly be stimulating their sodium-potassium pumps, or feeding it energy, if you will. So every time it uses an ATP, it's going to move 3 sodiums out of the cell and 2 potassiums into the cell. So if it keeps doing that, it produces a potassium gradient. So a lot of potassium inside the cell, little of it outside the cell. So this is just a schematic to help you remember this. So there's the potassium gradient. High potassium inside, low potassium outside. The opposite occurs with sodium. It's going to have a lot of sodium outside and a little bit of sodium inside. So that's the 2 ion gradients that have to be produced in order for a neuron to function. There's also an electrical difference. So notice neurons tend to have more negative ions on the inside and more positive ions on the outside. So this is the situation that has to be set up in order for a neuron to be able to produce an action potential. All right, so we have to make sure we remember that. The next step is to put it all together. Now look at this figure. This is a very complicated figure. We're going to start a new video because this one you want to kind of have it all by itself, because you might need to watch this next video a couple times to really get it down. But this next video coming up is going to be a home run video, meaning it's going to have a lot of information, and it might take a couple times through the video to have it make sense. So I'm going to stop this second video and start a nice new fresh video that's going to walk us through this slide.